Biological basis of autism spectrum disorder. Review of original research. Salience network-based classification and prediction of symptom severity in children with autism. Reviewed by Raul Mendoza. Background and purpose of study. Children with autism spectrum disorder either have hyperconnectivity or hypoconnected neural networks. Previous research has demonstrated that children with ASD have neural networks that are hyperconnected, which can explain the hypersensitivity some children with ASD may have towards lights and emotional reactions, as well as others with an astutely enabled memory. On the other hand, there seems to be some children whose neural networks are not well connected at all, which can explain why some children with ASD seem to have social deficits, difficulty with language, and impairments with other executive higher cognitive functioning. When a task analysis is conducted with children with ASD, some children who are suspected of hypoconnectivity tend to have problems doing more than one thing at a time, such as telling a story while drawing a memory. Some research on neural connectivity on children with ASD has deemed that it could be both hyper and hypoconnectivity, which could explain why some children with ASD present with more profound neurological problems, while others do not. Research has also demonstrated that early on in life, children with ASD seem to have slightly larger brains, and autopsies of children with ASD have revealed that they have more neurons than a typically developing child at the same age, which seems to suggest that the brain of children with ASD seems to develop in a different manner than typically developing children. What this means is that if there is a chance to finding out where the problem is occurring with regards to intrinsic functional connectivity, then research on the whole brain of younger children with ASD should help to lead us discovering what specific neural networks are being affected and how to better classify children with ASD with these scans. Experimental hypothesis. The authors proposed to find a way that neuroimaging could help differentiate children with ASD from typically developing children. The authors postulated that the functional connectivity of children with ASD would either demonstrate a hyper or hypoconnectivity specific to the anterior insular cortex, anterior cingulate cortex, and anterior insula, or what makes up what neuroscientists refer to as the salience network. So why target the salience network? Well, because the anterior insular cortex and anterior cingulate cortex have been associated with linking external sensory information with one's internal state of being. Furthermore, the anterior insula is important because it helps facilitate the neural networks that are implicated with distinguishing what is important to the individual and external stimuli and the self. And those of us who work with children with ASD will know there tends to be a social quote-unquote disconnect associated with children with ASD and the people around them. So if there's going to be a link to differentiate typically developing children and those diagnosed with ASD, it would probably be here. Summary of basic methodology. The authors recruited participants from local hospitals and clinics in San Francisco area. They looked specifically for children between 7 and 12 years of age, and they matched pairs of children with ASD and typically developing children by gender age, and IQ. Specifically, they used a sample of 20 children with ASD and 20 typically developing children, again matched by gender, age, and IQ. Autism diagnosis via both the Autism Diagnostic Interview, revised, ADIR, and Autism Diagnostic Observation Schedule, ADOS, along with diagnosis by a clinical psychologist, was made. IQ scores were obtained via the Wechsler's abbreviated scales of intelligence and average 112.6 for children with autism, and again matched for the children who were typically developing. The ADIR average scale scores were 20.4 for the social scale score, 15.9 for communication scale score, and 5.8 repetitive behavior scale score. For the ADOS, the average scores were 8.2 social scale score and 3.6 communication scale score. This is important in that just at face value, these children with ASD seem to be rather high functioning for most of the children that I have worked with in my experience. Instruments. A functional MRI was used. 6-minute scans were employed, and instructions were participants were instructed to relax, please keep your eyes closed, but do not go to sleep. A structural MRI was used to examine the structures of the brain of all participants. The functional MRI is and was used to examine oxygenation flowing through the areas of the brain that they were examining. Statistical analysis. The SPM software, or statistical parametric mapping software, was used. SPM software construction and assessment of spatially extended statistical processes used to test hypotheses about functional imaging data. SPM software is designed for the analysis of brain imaging data sequences. The sequences can be a series of images from different cohorts or time series from the same subject. Dual regression was employed, as it is typically used when comparing large-scale brain networks between two or more groups. Most important results. In this study, children with ASD were deemed to have hyperconnectivity in a number of neural networks, including the motor network, the motor neural network. According to Wenning, Norris, Dolick, Mew, and Calabrese, motor neurons are associated with movements, which could explain why some children with ASD could engage in characteristic self-stemming movements, such as rocking back and forth and shaking hands in the air. The visual network. Freeberg and Van den Bos described the visual neural network as being associated with seeing, which could explain why some children with ASD are sensitive to lights. The salience network was examined and described predominantly by these current researchers. The salience network is essentially the part of the brain that decides what is most deserving of our attention. Uden et al. 2013 described the network. This particular network has key nodes in the insular cortex and is critical for detecting behaviorally relevant stimuli and for coordinating the brain's neural resources in response to this stimuli. The insular cortex is complex and multi-purpose structure that plays a role in numerous cognitive functions related to perception, emotion, and interpersonal experience. Now, the frontotemporal neural networks are implicated in self-monitoring theory of mind, capabilities, processing/evaluation of internal mental states, perception of pain and emotions, and sustaining personality and the self. This could explain why some children with ASD may seem as if they are simply sitting there doing nothing, as if they are trapped in their own mind for long periods of time. Similarly, this could explain why some children with ASD are over-emotional in situations that do not seem to merit such reactions. The default mode neural network. This is a neural network that tends to be active when a typical person is not engaged/focused on any kind of task. Functional connections of this network include medial prefrontal cortex, medial parietal cortex, and medial temporal lobes. It is theorized that the default network is correlated to mind wandering and introspection, otherwise known as one's own stream of consciousness, which is never related to anything in an individual's external environment. Strengths of this study. Perhaps the biggest strength was the use of functional MRI and structural MRI, which means that one is literally peering inside the mind to see what is going on in there. Now, this is not to say that we're able to tell what each participant may be thinking, but what we can definitely say is with a high level of confidence that a particular area of the brain is being activated and hyperactive or hypoactive, and compare brain activity between the groups of children with ASD and typically developing children. Another strength is that this use of fMRI and MRI examined large networks of brain activity, whereas other previous researchers have examined just one particular structure or smaller areas of neural network connectivity. The final strength, main strength of this research was that using the functional and a structural MRI, it helped to understand and examine the integrity of the networks of the brain that were examined here. Now, weaknesses. There are a couple of different weaknesses that we found, that I found in this exact research. The biggest weakness was probably that they had a small sample size. A sample of 20 is, for the most part, a relatively small sample. Most of these children with ASD seem to have a very high IQ and higher functioning, which means that they were not a very diverse enough group of children with ASD, as children on the spectrum of functionality can range from very low functioning to very high functioning. And it looked like these particular sample of the population that they used of participants were very high with regards to functioning. Some of the older children were closer to adolescence, so neural networks may have changed. That is to say, that children that are younger would have been a better population to examine, given the previous literature. Another weakness was that these researchers did not control for comorbidities nor medications used. This information was not assessed nor collected. Significance of findings in the field of behavioral neuroscience. The biggest significant finding was that these researchers found specific neural networks that were implicated with autism spectrum disorder. Another interesting find was that children between 7 and 12 years old, at least in this sample, initially have hyperactivity in these specific neural networks that were mentioned. And later on in life, it leads to hypoactivity. This may be due to a neural correction of sorts to address overly stimulated areas in previous development. Another significant find was that neuroimaging scans of the neural network of younger children with ASD could lead to the origins of autism. This in and of itself was a good find through this research. Future research should be targeting a larger sample and a more diverse or representative sample of the population of children with autism. That means that we want to find, or we would like to include, research participants on the lower end, the middle, and the higher end of the functional spectrum. Another suggestion would be to use a younger group so that we can find where these neural networks are activated. And a last suggestion is to control for any medications or comorbidity to reduce any further explanations or confounds. Thank you.